Hi, everyone. Welcome to the P30 NIMH seminar today. It's my great pleasure and honor to introduce our speaker, Dr. Gendelman, who's the Margaret R. Larson Professor of Internal Medicine and Infectious Disease, Chairman of the Department of Pharmacology and Experimental Neuroscience, and Director of the Center for Neurodegenerative Disorders at the University of Nebraska Medical Center. Before joining Nebraska in 1993, he occupied various research positions and faculty positions at Johns Hopkins, the National Institute for Allergy and Infectious Diseases, the Uniformed Services University of Health Science Center, Walter Reed Army Institute of Research, as well as Henry Jackson Foundation. He retired from the U.S. Army with the rank of Lieutenant Colonel. Um, he has an exceptional track record. He has authored more than 600 peer-reviewed manuscripts, edited 12 books and monographs, hold 132 patents. He is involved in co-developing ultra-long antiretroviral medicines and in demonstrating the role of brain immunity in neural cell damage. The consequent pharmacologic and immune transformative therapeutics are aimed at preventing, treating, slowing, or reversing infectious and degenerative neural maladies. He's also known for demonstrating that AIDS dementia is a reversible metabolic encephalopathy. He's also a co-founder of Exavir Therapeutics. So and has won several accolades, which I'm not going to list here. If you want to see that, you can see it on his website. So without any delay, we'd like to hear from Dr. Gendelman about HIV infections and its effect on the progression of Alzheimer's disease. Thank you, Dr. Gendelman, for joining, and the stage is all yours.
Yeah, well, first of all, thank you. I hope everybody can hear me okay. It's certainly a pleasure and an honor, and especially to squeeze this topic in. I know the interest in engaging with this symposium is quite active, so I'm certainly appreciative. So we're going to really do something a little different today, and I want to start off with saying that this project is 25 years old. So I know you'll say, well, what have you been doing for the last 25 years? Well, we've been working, but we couldn't generate these animals until the CRISPR revolution actually occurred, that we were able to make real knock-ins of APP, PS1, and most recently tau. And we had to do it if we're going to be studying HIV infection in relationship to Alzheimer's disease. We had to do it on an immunodeficient background. Why did we need to do that? Because these animals needed to be infected with the virus, and the pathobiology of Alzheimer's, pretty much each of the aspects we're going to talk about in a little while, had to be reflected. So it was, again, the first 15 years of this project was pretty much a failure. We weren't able to put the pieces together. And I would say about a year ago, the project did come together. I will say one other disclaimer as we move forward. I know there's going to be interest. There's already been interest in the mice that we've generated. It'll be a couple months. We're not ready to, even though the first paper has been published, there's a series that are coming out on this model, and we really are nailing down key aspects of this process, and you'll hear about this as we move forward. Why is this important, and why did I push to give this symposia? Because the new combination antiretroviral therapy, including the long-acting and now the ultra-long-acting drugs, are allowing people to live almost pretty much a close normal life. So we're seeing patients in their 7th and 8th decades of life, and now there are beginning to be reports of what's happening in the later stages of HIV vis-a-vis the neurodegenerative diseases and the association, which to this point in time was due mostly to aging. Now, the reason for this slide, it did come from the pox vaccine early on, that people thought, my goodness, that if you take a vaccine, you're going to turn into a cow. That's where they came. And I know we're dealing with an era where there's a lot of changes in biomedical research and ideas of vaccination, ideas of generating chimeras, new animal models. In this case, we're going to be talking about a combination of a human glial brain, or a part of a human brain, in combination with a rapidly progressive Alzheimer's model. So a lot of things are going on, a lot of new ideas, new models, and new research. I want to end this introduction with one other point. This is a process in motion. This is actually the first humanized mouse or HIV infection of a humanized mouse that is a rapid progressor of Alzheimer's disease. So a lot of questions, and I hope you guys will ask a lot of questions because we're still dealing with this in a very aggressive manner. Just because we have a couple papers published does not mean we have all the questions answered. All right. Let's see if it works. Okay. So I just want to start off. I usually do this in the end, but clearly these project scientists are highlighted. This is not me working in the lab day and night. It's a group, people from immunologists, virologists, cell biologists, neuroscientists that have all worked together. We'll talk about names and associations at the end of the talk, but I'm really a very privileged guy because I've been in this field for 45 years. It's amazing. When I first came in the late, actually, it was 1981, when I first came to Hopkins. It's an amazing time when I was a postdoc with Dick Johnson and Bill Narayan. But a lot has happened since then. And I'm also incredibly lucky to be surrounded by a very talented group of investigators. Okay, so here's the question. Let's get down to basics here. Why are we doing this? Because it was a huge amount of work. The question we thought was important is whether HIV affects Alzheimer's and Parkinson's disease to develop in a differential way or more quickly. The clinical studies are still out. We still don't have definitive, I'm going to review those clinical studies in a moment, but the issues of HIV and Alzheimer's disease are something that we talked about for a very, very long time, that a person's risk for dementia would increase as they live with HIV. Why? Because they both have an inflammatory component. They both affect both the innate and the adaptive immune system. There is issues regarding plaques, tangles that can affect microglial responses, which are part and parcel of both disease complexes. And there's certainly reason from a pathobiological standpoint that there would be interactions between these diseases. The other problem and the other reason was that Alzheimer's transgenic mouse are limited. And we saw this 25 years ago when the first Alzheimer's vaccines were being developed. And as you know, and we all know, they were super successful in the transgenic models. But when moved into humans as the next case, there was a number of secondary comorbid problems, including edema. There was an encephalitis, meningoencephalitis, patients who had died subsequently when treated. And the Alzheimer's vaccine-related work really was stymied for quite a bit period of time. So why? Because they don't really represent the full spectrum of the pathologies. The neuronal loss is a minor part of these transgenic models. And the timing and the progression of the disease is not aligned. And the models, some of them focus on amyloid and plaques, but neglect other features that we talked about in neurodegeneration. So this was out there. It's been out there for many decades, but they're really only part, besides the abilities to study human immunity, they were severely limited by the overexpression of the transgene, which changed the pathobiology of the disease complex that was not reproduced in humans. So the other part of this came out recently within the last couple of years, and we're beginning. I'm not saying it's the end all or be all, because I'll get into trouble with this. These papers are just coming out. So the issues of people living beyond 80 years old with HIV and the prevalence that we know are still very limited in study and investigation. But for what we know in the few reports that have come out, and here's one of them, it's in abstract form. It's for the males, it's an increase from 26% to 33% in the 8th decade of life. For females, it's a bit more strident, 31 to 44. So there is an appreciable, at least beginning, that we're seeing that the incidence, prevalence, and severity of these neurodegenerative diseases, and again, we're just beginning to see them, or beginning to actually see these reports, are just coming out. So we all know what Alzheimer's disease is. I'll go through this incredibly quickly. It's the most common form of dementia. It's irreversible, progressive disease, destroys memory and thinking skills. But I think more important than just the pathobiological and clinical components of Alzheimer's are the number of people affected. And right now, there is really more than 7 million, 6 million is an older slide, but we're talking about 20, 30, 40 million people affected. So this is going to be even a bigger problem, a much bigger problem. And just a year and a half ago, just the GMP and the effect of our economy, we're talking about 345 billion for care, the effect of the patient, not only that, but the caregivers, the family, and we're talking about 145% increase in death. So this ain't going away, and we're going to be dealing with this much more frequently in the time ahead. Again, symptoms of confusion, difficulty remembering with familiar tasks, misplacing items, memory loss, difficulty solving problems. Again, this is something we all already know. The pathobiology, and we're going to get into this big time as we move forward, are the formation of the amyloid plaques, the neurofibrillary tangles, the paired helical, the hyperphosphorylated tau. Tau is not what we just published. We're about to publish a triple knock-in very soon that includes tau. But no question, these plaques that form, what we call amyloid plaques, are not only present, and many of us probably already have them in the 3rd, 4th, and 5th decade of life, but start inducing pathological features associated with aging, or what we think may be pushed in the presence of progressive HIV infection. So, and again, it's not simply a benign accumulation, but in some patients, there's all the secondary issues regarding what these plaques do to the innate and the adaptive immune system, what they do to neural communications, what they do to neurodegenerations and neurovitality. But again, this is really not the topic of my talk, but it's quite significant. You can look at the brain, the lower left-hand corner, the sulci, the change in the actual composition at the macroscopic level is also quite significant. Okay, so a model is needed to mimic, if we're going to study this and look at the progression of HIV infection, even in the face of antiretroviral drugs, how do we mimic this? How could we study this? How could we get a handle on not only understanding the process and the interactions, but also future therapeutics and how we're going to measure these differences as HIV patients continue to do well in the 7th, 8th, and even in the 9th decade of life. So let me just give you the bottom line in terms of what we've done. We've made an AD mice. In the first order, it was the amyloid precursor protein. These are knock-ins with PS1. We've also done this with tau. We'll again, talk about this in a moment. The knocked-in mutations were created by CRISPR technology. Why? We didn't want to have overexpression artifacts that were seen with the transgenic mice. We needed to do this on an immunocompromised NOG background. That was also critical. Why? Because inevitably we're HIV researchers, and we wanted to answer those vital questions of how HIV infection with and without antiretroviral therapy could ultimately affect the progression of the key elements of the pathobiological parts of Alzheimer's disease. Again, interested in adaptive and innate immune cells, we had to perform reconstitution, and we actually mated them with Dr. Garantlu's microglial mice. So they had a glial brain, a glial human brain, in the face of a human immune system, in the face of critical elements of progressive Alzheimer's disease. And again, microgliosis, astrogliosis, tau aggregates, in that case, formation or in the development towards bona fide plaques, the neurofibrillary tangles, which we'll talk briefly about. But again, we really needed the key features to be able to do this. And we do have a patent on this.
May I ask a question, Howie? It's Mary Jane.
Oh, how you doing?
I'm so glad to see this seminar. It's thrilling. Just give us a minute. Are these familial AD determinants?
Yes.
Thank you. Early onset.
Yeah. And we're in the midst. This is the first animal. We have another one. The UC Davis group in the mouse biology program is actually giving us a super amyloid we're doing additional mutations. So the disease is developed even more rapidly than what we have in the first line investigation. But again, with so much to cover, and I hope I get through this talk. I think there's a 50-50 chance I won't do it, but I'm going to try my best.
Thanks.
Okay, so this is why, and this is answers to your question, Mary Jane, that the FAD mutations were knocked in. These are the familiar forms. They're all driven, again, by the endogenous promoters, and they lack overexpression artifacts. We're not the first. Again, I'm not going to lie to you to say that we're the first to do this, but were done on an immunocompetent background, and there were single knock-ins or double knock-ins in one case, but no one to this date has done it with a human immune system, a partial human brain, glial elements, and the ability to study both innate and adaptive immunity with real human cells. So we're able, and this is what was new, is what we were able to overcome the immunocompetent background with limited testing, but we now have that in tow. So again, I'm going to make sure I get it before I go on, because it is complicated. We got all the knock-ins, the principal genes, in a NOG background, the immunodeficient background, with a human immune system that we can use these mice for infection with HIV or treatment of antiretroviral therapy. Okay, so this really goes back to Mary Jane again. I'm not trying to answer your question 4 times, but this was not done on purpose. This was done as a natural progression, but these are the creation founder lines. Again, both the APP, the PS1, this is the location where in the gene segments that converted from, because we did knock-ins, from a murine to a human system. These were all knocked in each of these homologous genes. They're, again, driven by endogenous promoters. And I know I said this 5 times, but this is a big deal. There was no overexpression artifacts. And that was the big problem with the bona fide transgenic AD models. So we were able to overcome that limitation. Okay, so let's go into some of the pathology that we're seeing. This is the knock-in mice. So we do see intraneuronal amyloid beta and microgliosis, and we did get into a lot of questions. So let me just give you the question that you're going to ask me, and I'm gonna jumpstart that. The plaques, the actual bona fide plaques, are plus minus. And I know if you read our paper, it may be a little confusing because people said, do we really see plaques or do we not see plaques? We did see plaques, bona fide plaques, but we have to extend it beyond 16 months. So you know how long these animals live and how long it takes to finish these projects. So we're still working on the plaques. We believe that the plaques are there, and we have seen it, but I would say at this pace, we did say plus in our paper, and we did have a neuropathologist look at these, but the plaques are still early. They're early. But A beta is seen intraneuronally, so we do see accumulation of A beta within neuronal cells and cell bodies. We see a very robust microglial reaction. You can see the merge in the lower corner, so there is the microgliosis associated with these intraneuronal inclusions. And when you look, at least in a dual piece, in the NAPS, and the NAPS are the ones that have at least 2 mutations compared to the NA, which is just amyloid beta alone. There's no question there's a profound innate immune response. We're going to get into this and dissect this a little bit more. But the bottom line is we were able to show intraneuronal A beta inclusions. We were able to show a microglial response, and they were related one with the other anatomically. All right, does everybody follow this? I hope so. Silence is always a problem. Okay, but this is a big deal. I think this is a big deal for us. But we were really excited about this when we got it because all the previous models were not able to reproduce any neurodegeneration. And in the NOG background, we were able to see progressive neurodegeneration over time. The entopic human beta deposit in the knock-ins really cause widespread neuronal loss. And this is a big deal. I believe it's a very big deal for this model, unlike the models that are out there, the transgenic models. And it's pretty obvious if you look at these slides. You can actually see on the single knock-in compared to the double and now the triple knock-ins, where you can see very profound neuronal or neurodegeneration, and in the hippocampus, in the cortex, and we actually start seeing it at 6 months. So it's relatively early, and the more the increase in the numbers of knock-ins makes the degeneration obviously much more significant. All right, everybody follow me so far? I hope so, because it's going to get a hell of a lot more complicated. So I just want to make sure the basics are there. Okay, the other part of this is we began to image these animals. This is a demonstration of brain atrophy. So there is, when we looked at the right and left ventricle and the cortex, and in the beginnings of our cognitive testing, which is still in its infancy because we have just very few animals that really, this is early data, I would say. But what we can say for certain is that there is atrophy. There is neuronal degeneration and atrophy that we can pick up from these animals by a high field MRI examination. We're still working on nailing this down, and the problem, as you can see, with the novel object and the Y-maze testing, these are immunocompromised animals. So we can't do a Morris water maze. We're limited with the number of behavioral tests we can do. And if you look at the NOG animals, you can see very clearly the splay is quite significant in the controls. So the only really evaluations can be done based on time and aging. And this is something that we hope to overcome as we investigate more tests and greater numbers of animals. So it's still early in the game, with regards to behavioral testing. Okay, so this is the summary on the first part. This is certainly a bridge to AD. The transgenic models show overexpression, based artifacts, the immune system is a mouse, the microglial response is a mouse, and the neuronal degeneration is extraordinarily limited. And that's where we got into problems with the early testing of vaccines for Alzheimer's disease. These knock-in models, the ones that we developed here, are driven by indigenous promoters. They mimic the clinical AB deposition patterns. There's relevant disease progression. They're constituted or reconstituted with the human immune system, and they can be infected with HIV, and I'll show you this in a moment. But most importantly of this model, I believe that is most importantly, is the significant, very significant neuronal loss and brain atrophy. This is a very new finding with any mouse model of Alzheimer's disease. Okay, so can it work for both diseases? And this paper just came out. We got excited about it. I think most of you probably saw it. It came out in Nature Com a month or two ago. But they said that HIV-associated neurocognitive disorder and viral reservoirs obviously are a challenge. There's no question in trying to mimic this for an aging model. But this paper actually showed quite convincingly that both alpha-synuclein for Parkinson's and amyloid beta fibrils did enhance HIV entry and replication. The amyloid beta fibrils did promote virion attachment and cell fusion with the virus. So this was the early evidence published in a reasonable journal, in Nature Com, that there was the potential for an interplay between HIV and brain amyloids, and maybe a contribution in the later stages in the aging process, that HIV could affect some of the pathobiological features of these neurodegenerative diseases. Okay, so we needed to test this, and this paper came out, and I'm going to give a little swing for a new journal because I'm actually the president, and I guess this is a disclaimer of the Society for Neuroimmune Pharmacology. This is the journal of the society, and this paper, I guess, formally will be out on Friday. So really, will be out very soon in a formal way, although it can be gotten from the website as well. But what we did with the help of others is we took this mouse and we generated a hybrid between what we've had and Dr. Polektova and Dr. Garantla had developed for several years, that we were able to mate this mouse and generate an Alzheimer's microglial mouse. So again, the brain has human-like microglia in the face of these rapid mutations of Alzheimer's disease in an immunocompromised background. So we were able to begin to study in an early report. It's not definitive, it's a first paper, it's a brief report. We understand there's a lot more to do. But what we were able to show, what we were excited about, is that HIV infection does in fact increase the brain amyloid beta load. We were able to demonstrate productive infection. We were able to measure both soluble and insoluble A beta. We were able to show these results reproducibly, and they were statistically significant. The other thing that we saw is that the DNA, HIV DNA in tissues, which both in spleen and brain, you can see the P24 stains on the left in spleen, HLA-DR, it's a human HLA-DR for HIV, and the control, there was really no difference. But the P24, you can see easily in the spleen. You can see clearly the pathobiological hallmark of the myeloid infiltration. But we were very excited to see that we were able, for the first time in these Alzheimer's mice, to see productive HIV infection, both in the cortex and the hippocampus, in the lower fragments, or these these pictures showing the HIV P24 staining, which were quite robust. So easily infectable animals, a significant level of increase in soluble and insoluble A beta. And we did have the system now in play to begin to study these diseases in context, not complex, in context, one with the other. And when we look further on the crossroads of this neuro-HIV and Alzheimer's disease, we can see a clear association between the amyloid beta fibrils and the activation of the microglia. So in the bottom part of the HIV infected in C, in panel C, you can see the activated myeloid cell in the face of the fibrils that are forming. And when we actually looked and did the quantitation, you can clearly see increases in both soluble and insoluble amyloid beta 42. So we're beginning to show that these diseases may intersect one with the other. But we didn't stop there. This paper has not come out, and mainly a very, very talented young graduate student. We're trying to get him to work. Unfortunately, he decided to get married, so we lost a little bit of time. No, I didn't say unfortunately. Marriage is a good thing. That has nothing to do with this talk. That was supposed to be a joke. But he did get married. He just came back, and hopefully we'll finish this. But we needed to really nail down the association between the plaques, both intra and extracellular, and the key features of Alzheimer's disease. And the only way we could really do this is by spatial transcriptomics. So we're really not going to have time to go into this. This was tremendous, tremendous amount of work. It's almost finished to the point where we'll be able to write it up. But what we were able to do is begin to do selections in different regions of the brain and begin to look at regional changes in the transcriptomic profiles and base it on microglial, astrocyte, neuron, and nuclear systems or stains, where we can begin to really understand cell-cell interactions, regional interactions, degenerative interactions that affect the transcriptomic profiles that we're seeing in a very complex animal system. And this gives you kind of a highlight of what we're looking at in the photo changes, but we now are able very nicely to begin to look at regions where there's no amyloid beta is present, amyloid beta and HIV, evidence for HIV with proper controls as we move forward. But I think what we can leave with this, and this is going to be a huge amount of work. It already is a huge amount of work, but when we write it up, it's going to be a very, very complex paper. But what we're seeing, interestingly enough, if you look at the changes, we were expecting to see most of the action in the microglia, obviously the target for HIV. That's not what we saw. Actually, most of what we're seeing between amyloid and HIV in the progressive form of Alzheimer's disease was in the neuron. And that was something really robust and exciting to look at the changes in neuronal responses during these dual infections. And we begin to look at the key genes that are upregulated. These are the pathways. But I think what really is interesting is when we're examining these particular genes and gene processes and really getting into the in depth of understanding pathways, which we are doing now. What is really coming out is, and very clearly, is there's overlap, that what we know is affected by HIV in the brain, and we know what is affected by amyloid, or these plaques, as well. And when we put them together, we're clearly accentuating these pathways that are linked to the progression of both diseases. So this is something that we're, again, quite excited about. but we have a long way to go to look at cause and effect, especially with such a robust neuronal response that we're seeing during the dual HIV and Alzheimer's model systems. And again, I'm going to stop. I'm only going to show you one more slide, and these are just names of pathways. But again, where we're seeing the changes is mainly associated neuronal pathology. So all of this, both the virus and the amyloid, are kind of being super focused on the neuron, which is, again, something that we didn't think about. We thought that it was all going to be on the glial, at least for the most part, in the microglia, but that was not the case. The accelerated neuronal pathology appeared to be a major component of this process. Now I'm going to show one slide on this because I get into a lot of trouble with this. So you may throw stuff at my screen, but I want to go into this. And I'm going to tell you something that about me personally that you're going to laugh, but I'm going to say it easy. We believe that we can generate a mouse that can be HIV infected. Now, you're going to look at me and you're going to say, Dr. Gendelman, you're all full of it. There are problems with the receptor. There's problems with the signaling. There's problems with processing. There's problems with maturation. You're not going to be able to do it. It's a ridiculous project. And trying to move this into a natural mouse that has a natural progression of Alzheimer's disease can never happen. So I said, okay, but let's try. Let's try. So we began and we worked with kind of the best mouse biology program on the planet, which is in UC Davis. Yeah, everybody knows about that. And we convinced them to work with us. They convinced us to work with them. I think it was a bilateral issue. But we're beginning to dissect a multi-class animal that has knock-ins for all the signaling pathways that are relevant. Obviously, CD4, CCR5, some of the mitochondrial key elements that are known to be affected in a mouse system that would restrict HIV replication. But the most important part of this, and I'm not worried that somebody's going to steal our ideas because we've been working on this for so many years. To put each individual knock-in together is basically a half decade of work. So I'm going to share it because maybe you guys will have even more ideas to help us. But I think we've got it close to being nailed down. And I think we're able to generate a mouse chimera for CD4 and CCR5. So the receptors are correct. We were able to muck the complement receptors, try to affect some of the signaling pathways. But the big problem that we had was the maturation. So as you know, in immunology and T-cell development, there's a precursor cell that's a chimera between CD4 and CD8. And that was a big problem for restricting HIV infection in a mouse. So we needed to get a human MHC class 2 to help in the maturation of CD4 and CD8. We're very close to this. We don't have it yet, but we're very close. And these are some of the experiments that are ongoing. Probably take us another year or two, but I'm still confident that we can get there. Do I have it? No. Are we working on it? Yes. And I think the Chen Zhang, she's an unbelievably successful postdoc, and Prasant Dash, and has been a big help, and Lee Mosley. We have a lot of people involved that are Larissa as well to try to get this to the finish line. So this is the summary of the pathobiology of the two. And this, again, are these key elements. I stole this from our friends in San Diego in their recent Nature reviews in neurology. Ron, I hope if you're in the audience, I hope I didn't I hope I've done a reasonable job in summarizing your paper, your review. But I think we all know this. These are key elements of NeuroHIV, that HIV infects myeloid cells. I think that's been known for 35 years or longer. Microglia do serve as a latent reservoir, even after antiretroviral therapy. That activation of microglia and perivascular macrophages leads to pro-inflammatory cytokines and neurotoxins as well. It's controlled in large measure by antiretroviral therapy, but still produced at low levels. And that's why up to 40–50% of people still develop cognitive dysfunction associated with HIV infection, even with drugs or proper drugs treated. Astrocytes respond. We know about that. And we see that neuroinflammation leads to neurodysfunction. So amyloid in the face of inflammatory response forms these fibrils, and they become part of this disease complex that I just shared briefly with you. Okay, I got 20 minutes, and I want to go to the last part of my talk. Are there any questions on the first part? Okay, everybody understands everything. That's good. So you can teach me, because most of this I don't understand either. So really where we're going with this is new therapeutics. So we're going to talk for 15 minutes on what we're doing with therapeutics. And many of you know already that we have clinical trials that are ongoing with drugs that will generate immune transformation from effector T cells to regulatory cells, working on getting these regulatory cells into the brain. We're looking at neuroprotection in balancing the TREG cells, which are immunosuppressive and could make a number of diseases worse, not just HIV, but obviously cancer and autoimmunity can be affected as well. So getting these cells into the brain and affecting the disease complex is something that we've been working on for a bunch of years. And actually, we were about to publish our first phase, our third phase 1 trial, and we finally raised the money to do a phase 2. We ran out of money. We wanted to have a larger patient population. So I'm very proud to say that we're very close to an agreement with Saudi Arabia. They're going to help us do this study. And you're going to ask, what is a Jew, a New York Jew working in Saudi Arabia? That may be a question, but I think that's fun. I think if we can set up interactions and dialogues and collaborations throughout the world, regardless of who we are, what our background is, I think that's a super addition to this research. So I'm looking forward to spending time in these countries, and I think only good will come of it. So it's really exciting. So I think the first thing that we saw is that Coya Therapeutics, and we licensed some of these drugs and our patents to Coya. So these are next generation of using ways to enhance regulatory T cells. These have been open label for Alzheimer's disease and Parkinson's disease. And preliminarily, they look quite safe. This is IL-2 derived regulatory T cells. We've done this with sergramestin, other pharmacological agents, and we see some hints. Again, we're not going to be able to see any affirmation of clinical benefit until we complete our phase 2/3 study. So we got a ways to go. The idea, though, overall of where we're going with this program is generate amyloid beta specific TREG cell therapy. So what the heck are we doing? Well, when we did our IL-2 or sergramestin or whatever, obviously, if you look at the adoptive transfer, the blue dots are neuroprotective regulatory cells, and only a small fraction actually enter the brain where the disease is. This is an Alzheimer's example, could be Parkinson's, ALS, it doesn't make a difference, but only a small percentage of these neuroprotective cells will actually get into areas of injury. So we really had to change the MHC, the immunological complex, so to speak. So they would significantly or only react to the amyloid beta and specifically to the fibrils. So we were able to isolate these neuroprotective regulatory cells. We were able to engineer in the MHC complex and then make them recognize specifically, as you can see below, the amyloid beta or the plaques. And that's where they would travel across the blood-brain barrier and ultimately affect the clinical response. And you can see that we published this about a year ago, but it's compared to polyclonal regulatory T cells, and this is the pathology, the neuronal dropout, the microgliosis, the astrogliosis, the accumulation of plaques in the face of inflammation. We were able to show that these antigen-specific neuroprotective regulatory T cells will migrate and accumulate to sites of cognate antigen, and they will be immunosuppressive, knock out the inflammatory response, and then be, in fact, neuroprotective. We were able to show increased efficacy, and we were able also in the brain to reduce immunosuppression and secondary complications. And again, this is all published work. The AB-specific TCR, the T cell receptor, again, I don't have time to go into this, but these are T cell antigen specificity is attributed through the MHC, through the T cell receptor. And these antigen-specific TEPs are where we started because they're much more common in blood. And then we were able to transform them and then generate an immunization scheme from actually patients where we did this both in human and in mouse cells. And we filed a bunch of patents on this. We published a bunch of papers showing that the effector cells treated like this that are antigen specific will exacerbate Alzheimer's disease, and the amyloid beta TREGs, the neuroprotective, anti-inflammatory, immunosuppressive TREGs will ameliorate amyloid, the disease complex. And this is all through the TCR receptor. And we showed this clearly pathologically, that the amyloid beta TEPs will accelerate the AD pathologies, more microgliosis, more neuronal dropout, more degeneration, very clear. When we generate our specific TCR amyloid beta TREGs, they were difficult to transduce. We were able to get through this experiment by serial adoptive transfers and electroporation, but ultimately through genetic engineering, expression, transfer, and transformation, we were able to use CRISPR technology, again, to augment the amyloid beta specific TREG. This paper was published a year or so ago and demonstrating proof of concept. Okay, we showed that they can home into the brain. As you can see here, these are using a standard APP-PS1 mouse, not our more recent mouse. They get into the brain very quickly, within minutes, and they're sustained. Very different than the TREGs, the polyclonal TREGs, which don't get in very easily. But you can see they clearly accumulate. They're still functionally active, immunosuppressive, and that's clear by all the assays that we've done to look at suppression of effector cell responses and neuroprotective outcomes. We were able to show all of this. And then the most important, which I will spend a minute going on, is we were able to show that these specific in pink in the amyloid beta TREGs show a decrease in the amyloid burden, a decrease in the thioflavin, the dense plaques, a marked decrease in the microgliosis or the microglial responses. All of this is done quantitatively. We've seen it in the cortex and the hippocampus, and we were able to show that these animals actually develop improved cognitive function. So big change in pathology, big change in homing, big change in the pathobiological responses, big change in inflammation, and improvement in cognitive function. And this is all part of our Y-maze test. And we were able to do this with water maze because these are the regular mice. But if you look at the block 3 response, that those animals treated adoptively transferred with polyclonal TREG here compared to our transformed A-beta specific TREGs, a big difference in cognitive function on the 3rd block of testing. Okay, all these therapies are gaining traction. Obviously, we hope to bring this to humans. This is extending therapies. Most of the CAR TREGs are developed for solid tumors. The massive technological advancement in CAR TREG therapies is in managing adverse events, and we're really working hard to generate these cells. Autoimmune disease is the frontier of this new TREG therapy, the CAR TREG, and we're not the only game in town to look at this, but we are the only game in town to move this forward for neurodegenerative disease. And this is the CAR TREG MHC independent recognition. Again, we don't really have time to get into this, but as I said, these are MHC restricted. We've done through the T cell receptor and the peptide and the MHC-1 that we can ultimately generate these T cells. We're working on generating them in relatively high quantities that we can begin to do both preclinical and hope not so distant future clinical studies as well with the CAR TREG therapy. Okay, so this is where we are, and I'm going to try to finish up. The CAR TREG development stages. We've now developed antigen target identification. We've developed and we've characterized these, and I've showed some of the biological and preclinical evaluations. We've developed a CAR plasmid design and our vector development. We've transduced, and we're beginning, we don't have it all yet, to develop a CAR TREG therapy that could ultimately be used in human testing. We had to develop antibodies for the CAR component of this, and the FDA drawbacks. All of the works that are on just with the antibodies alone have only been moderately effective. The reason is poor specificity, pathological AB forms, and clearance. There's also secondary problems with drugs that are either in motion or have been used for amyloid-related imaging abnormalities, and the detrimental role of antibody-mediated TEF responses is still unclear. These are the 3 products, the 3 antibodies that are developed by others. Ours is called 2CD3M, which will, in fact, not recognize the monomer, which is the normal part of the AB or amyloid beta, but they will recognize the oligomer and the fibrils. So we do have a big advantage. And the question is, how did we do this? This is boxed in red. We've been able to do this through the advent of artificial intelligence. And rather than screening tens of thousands of antibodies, we were able to use specific peptides that will target just the pathogenic forms of Alzheimer's disease in this development. These are the neurotoxic A-beta isoforms. And again, and this will be near a conclusion here, we've been able to identify a unique epitope of the amyloid beta oligomer. Part of the antigens recognized, and this was all done by our colleagues in the UK. We didn't do any of the artificial intelligence recordings and development, but this was a major, major help for epitope identification in designing what we're going to be looking at, what we're going to be propagating in the antibody screenings that we've had in develop. This is how we develop the antibodies. This is how we tested and screened the antibodies. But I think this is the most important, is basically showing a comparison of the aducanumab compared to what we call our in-house antibodies to get us to the finish line of CAR-Treg therapy. We're showing a paucity of reaction to the monomers, but a very strong reaction to the oligomers and the fibrils of amyloid beta, which is a big, big advantage from what we're developing. And our antibodies do recognize what we need to have them recognize. These are hybridoma antibodies compared to the commercial antibodies, but you can see what we've developed here in Nebraska is equivalent. Okay. This is our CAR and vector design to finalize this, which I have no time to finish, but this is the summary now. So obviously there's a need to develop treatment for neurodegenerative disease. It's a Alzheimer's disease therapy is unmet. The amyloid beta vaccine and antibodies that our FDA approves are kind of plus minus. Why? They trigger an effective T cell response and meningoencephalitis, and the amyloid-related imaging abnormalities also present. Clinical trials are ongoing for TREG expansion, and low-dose IL-2. We're involved in that as well. Some of this data has just been published in the last couple of years. And then hopefully we'll have put these pieces together now that we have the models, we have the antibodies, we have the cells, and we have enough of the preliminary data that the hope is that we can move this forward to therapies. Okay, so this is my summary slide, and I got 2 more minutes. We published proof of concept for A-beta specific TREG therapy. We've completed the milestones in CAR-T development. We've developed the antibodies against the neurotoxic amyloid beta isoforms. We have the CAR vectors in hand. We're now enabled to expand this both in Alzheimer's and Parkinson's. And most importantly, and I spent a lot of time in the beginning, is we have the models. We now have in our hands the models that can be used for development, testing, proof of concept that we hope will aggressively allow us to move these therapies, these ideas from the laboratory bench and ultimately for human testing. And again, this is not my work. I was a cheerleader, a coach, a supporter, but all of this was done by skilled scientists, investigators, thoughtful researchers, I would say friends, collaborators in the lab. I can't mention everyone because I'm basically out of time, but the key elements are Pravin Urpuri, hopefully he's listening, Lee Mosley, who became a lot of the concept, hope, and development of this idea to new and advents of preclinical development. Yaman Liu, who now manages over 800 of these animals and tries to keep track of all this. Shorav, who I told you just got married, we should congratulate him. He's a senior graduate student who did all the, and finishing all the topical transcriptomic analysis. Susmita, who's done all the biomarker work. Goku, who's done and beginning to do all the migratory work. Rana, who came up with the co-therapies, both the cellular and the CAR-T. She's the first to develop proof of concept for this. Emma Foster, who's really been the linchpin of putting all this together. Amiko, who was with us in some of the basic cell biology. Chen, who's really developing this next phase of direct HIV infection. Santi Garantla, who is our animal model guru. Prasanta Dash, who's done a lot of very, very sophisticated developmental work for the LD. Jayton, who conceptually started all this work. Mohit, who generated data with antiretroviral therapy. So if I missed anybody, you'll have to shoot me, and I think I'm going to stop. I'm exactly on time.
Thank you so much, Dr. Gandelman, for a very nice talk. We have basically run out of time, but if we can take 1 or 2 questions at the max, if you still have a few minutes, Dr. Gandelman.
Yeah, I'm fine. I'm the one that went over, so go. Please, if anyone has any questions.
Matt, I see you have a question in the chat. Do you want to ask that question?
Yeah, I just wanted you to clarify the NOG knock-in mice that you used. Are they immunocompromised? Do they have no immune system, or did you reconstitute? Do they have the human immune system? Those mice specifically where you saw the neuronal loss.
They were not reconstituted. The ones that we just published, that paper, they were not reconstituted.
And so how does that, David Holtzman a couple years ago published a paper in Nature with a tauopathy mouse model, so a little bit different. But his argument is really that neuronal loss is driven by the cytotoxic T cell response. He sees a very strong T cell response that impairs synaptogenesis. But you're not seeing any neurodegeneration. You're seeing a lot of neurodegeneration in the absence of an immune system, in the absence of T cells.
Yeah, and I just say that the animals that we use and the controls for the HIV experiments obviously do have a reconstituted human immune system. First of all, there's absolutely no question, and we studied it along with many other investigators, that CD8, CD4 effector cells will drive neurodegeneration. There's no question about that. I think what was interesting and unexpected, and there has been just a few papers on this topic, is that the NOG background itself in the face of these Alzheimer's genes will generate neurodegeneration, will generate some of the loss of integral tissue or brain tissue that we saw in these animals. So we're very anxious. We haven't done it yet, but we are in the midst of setting these experiments up for what we feel will be, when we reconstitute these animals, when we looked at effector responses, when we evaluate the degenerative activities, we'll be able to even accelerate the neurodegeneration even more. But clearly, we're seeing it de novo from these animals in the face of these knock-ins of APP and PS1. So those are the results that we're seeing, and very reproducible.
Thank you.
Thank you again, Dr. Gendelman. I think in the interest of time, we'll wrap up here. Thank you for a great talk, and hopefully if more people have questions, they can reach out to you via email. Thanks.
Yeah, no problem. And Mary Jane, before I leave, my last thing, please, we haven't talked in a long time, but I know we have a grant together, so I hope we'll be able to continue this dialogue in the future. That's all.
Howie, there are so many questions that this magnificent work prompts. Let's talk. It's stunning.
Okay, well, thank you very much. You've been a friend for I don't know how many decades, so we'll keep talking. Thank you. Everybody have a great day. Thanks so much. Okay, bye.